This is Dr. Donald Leung, Editor-in-Chief of the JACI. Joining me today is Dr. Max Seibold, a scientist at the Integrated Center for Genes, Environment, and Health at National Jewish Health in Denver, and an assistant professor at the University of Colorado. I am delighted to have the opportunity to discuss with Dr. Seibold his article in the March 2014 issue of JACI entitled Dissecting Childhood Asthma with Nasal Transcriptomics Distinguishes Subphenotypes of Disease. Dr. Seibold, can you tell us what was the scientific problem that you wish to tackle?
Sure. So in this work, what we were trying to do is see how closely the nasal airway epithelium correlated with the bronchial airway epithelium in terms of gene expression. Since the bronchial epithelium is definitely an integral part of the development of asthma, it's a key tissue that we would want to analyze, especially for gene expression analysis to try to phenotype asthmatics. Unfortunately, to collect that tissue requires the performance of a bronchoscopy, which is a fairly invasive procedure. And we were interested in whether we could sample a much more peripheral tissue, the nasal airway epithelium, and see if we could see the same patterns of gene expression that have previously been seen in asthmatics' bronchial airway epithelium.
And what were your results?
So our results were, we saw a tremendous overlap between the expression pattern, and this was done by whole transcriptome sequencing, between the nasal and the bronchial airway epithelium. About 90% of the transcripts expressed in the bronchial epithelium are also expressed in the nasal airway epithelium. And in fact, we see a very strong correlation, 0.87 correlation coefficient between the expression of those overlapping genes between the tissues. And the second part of the study, once we saw that there was large overlap between the tissues, was to see, looking at a large population of asthmatics, 100 childhood asthmatics, 50 childhood asthmatics and 50 controls, and see how the nasal expression differentiated those two groups, and then look within those asthmatic subjects and see if we could subphenotype the asthmatics or if they had different expression patterns based on their disease.
What were the genes that you found to be most different?
So, talking about the genes that were differentially expressed between the tissues, I think the first gene you have to look at, and it's a gene that's very familiar with people studying asthma, is IL-13. When we looked at 100 genes in our targeted RNA-seq analysis, we saw 48 of those genes were differentially expressed. But when we looked at the correlation between the IL-13 levels that we measured and those other 47 genes, we found that the majority of those genes were strongly correlated with the expression of IL-13. And this showed to us that a lot of the driving force behind the expression pattern in the airway epithelium is probably driven by IL-13 and the underlying Th2 inflammation.
Were there any surprises?
Sure. So I would say first off, the fact that we could measure IL-13 was a surprise. We used a new technique that's based on next-gen sequencing, enriching the RNA from the samples, and that allowed us to measure for one of the first times IL-13 expression levels in the airway. And seeing IL-13 levels in vivo at the site of the tissue allowed us to make these correlations between epithelial genes that previously you could only establish those relationships in an in vitro manner. In addition to that, we picked up other immune genes like mast cell genes. In fact, CPA3, which has been shown to be up in the bronchial airway epithelium and is really a marker of mast cells, we saw was up. And I don't mean to say that there wasn't other non-atopic or non-Th2 inflammation genes found. In fact, we found several genes that were not correlated with IL-13 levels. And these happen to be genes that were markers of specific airway epithelial cells. In particular, we saw keratin 5, which is a marker of basal airway epithelial cells, was differentially expressed in asthmatics. In addition to that, we saw MUC5B, which wasn't associated with IL-13 levels, but was differentially expressed in asthma and not with atopy. Again, that's a marker of a secretory cell. So it seemed that we were maybe picking up also a signature of differential expression in the airway epithelium that's driven more by remodeling of the airway as opposed to inflammation.
Which of these gene expression changes were unique to asthma and not likely to be found in the skin or the gut?
Right. So I think it's difficult to say which of these would be unique to asthma. Besides the ones that were not associated with atopy, and those were the ones I was mentioning, MUC5B, keratin 5, there was 5 genes that we found that were differentially expressed only in asthmatics and not in atopic subjects. I think in general what we see with our results, seeing a large overlap in the same differentially expressed genes in the bronchial epithelium and the nasal epithelium, is that there's really a systemic inflammation that's interacting with the epithelium and driving most of this differential expression.
What do you think is the significance of your findings for our audience?
So I think our findings have significance both for the research community and for the clinical community. In particular, the ability to subphenotype asthmatics, as we were able to do in this paper by Th2 high and Th2 low, is a very important advancement, or to be able to do this in a way that doesn't require a bronchoscopy. If you suppose that these subjects have a different etiology behind their disease, then if you can subphenotype lots of patients in a research study, you can have more valid findings because you're able to establish more significance. And that's especially important in genetic studies where we know there's a lot of heterogeneity in asthma, probably heterogeneity that extends to the etiology of disease. And if we can separate out those patients in numbers that we need to, for instance, do genetic studies, then we can look at the genetic or find the genetic determinants of those subphenotypes and really advance the research field. In terms of the significance to the clinical community, we saw that the Th2 nasal pattern was associated with much greater risk of not just asthma, but atopic asthma, rhinitis, high blood eosinophils. And we found that nasal IL-13 levels were a strong predictor of whether the asthmatic had experienced an exacerbation of their asthma in the past year. So this potentially gives us clinicians a way that they can sample, and the sampling procedure takes really only a minute to brush out the cells, and from the expression profile potentially predict whether an asthmatic might exacerbate or not, and maybe you would tailor their therapy based on that. Additionally, I'll mention that since we found the same Th2 high and Th2 low patterns that have been seen in the bronchial airways, those patterns were previously associated with corticosteroid response. So again, there's the potential that the nasal expression profile could be used to decide whether to put a patient on steroids or not. Of course, these are research findings, so this would have to be validated.
And what are the limitations of your study?
So I would say one of the limitations of the study is this is a retrospective cross-sectional cohort. So despite the significance of our findings, these findings will need to be replicated and, if possible, repeated in a prospective manner. Another limitation is we just had a single point measure or a single brush from these patients. And it would be nice to do longitudinal studies going forward, trying to see the stability of these nasal expression patterns. But since we saw their association with phenotypes that are known to be stable, like whether you have asthma or whether you're atopic, we shouldn't see those associations unless the pattern was fairly stable.
Well, Dr. Seibold, thank you for joining me today to discuss these very interesting findings, and we look forward to hearing more about your studies in the future.